Close your eyes for a second and imagine two small, powerful hubs of sensation tucked into very different parts of the body. One is often spoken about with jokes and nicknames. The other hides under language and shame. Yet it is the single most densely innervated human organ. Welcome to a quick sharp trek through the biology and culture of your stories and Roman manship. The penis and the clitoris, two cousins from the same embryonic family. Two keys to human pleasure, and two brilliant subjects for serious research. Start with birth. In the embryo, a single tissue fold can become a penis or a clitoris. That means they share DNA, the same basic scaffolding, and surprising homology in erectile tissue. Say it like a biologist. Same roots, different branches. That simple fact explodes myths about fixed gendered function. And it puts anatomy at the center of curiosity. The penis has a visible shaft, a glans with a corona, and nerves that map to the spinal cord and brain. The clitoris has a visible glans too, but most of it is internal. A body, too, crura and bulbs that hug the pelvic bones. You cannot truly appreciate either organ without imagining beneath the surface. Neuroscience adds drama. Millions of sensory nerve endings send fast, precise signals to the somatosensory cortex. The brain decides what counts as pleasure. Hormones to the system. Testosterone, estrogen, oxytocin, dopamine, hormones that make neural circuits more or less ready to respond. Functional imaging studies show overlapping but distinct brain patterns when people report sexual pleasure. That means context, mood, safety, memories, matters almost as much as touch. Now the social plot twist. Language shapes experience. Nicknames like chin-chin or clit can make bodies feel playful or shameful. Cultural histories map modesty and taboo onto anatomy. And those maps change how people learn about their own body. That's where you, as a curious student, step in. You can be a scientist and an advocate. You can study textbooks, but also listen to real stories. Mixed method research combines surveys, ethnography, and lab measure. And you begin to see patterns of learning. Shame, pleasure, and health. Let's be practical, ethically practical. If you want to study sensitivity, you can use quantitative sensory testing in the lab. Use calibrated touch, safe vibration devices, and psychophysical scaling. Never exploitative experiment. You can model internal anatomy with MRI and ultrasound. You can run fMRI studies to see the brain's response. You can map variability, size, shape, innervation, and how these relate to reported sensation. You can investigate how education, consent culture, and sexual scripts shape feelings and behavior. Safety and consent are non-negotiable policies. Any research with human participants needs clear consent, privacy protections, and trauma-informed design. If your curiosity includes clinical practice, know STI prevention basics, hygiene, and when to refer to specialists. If you teach craft language that is precise, inclusive, and devoid of shame. End with a challenge. Become the kind of scholar who asks surprising questions. Why does the clitoris remain absent from many anatomy atlases? How do early cultural messages rewire nerve pathways for pleasure? How do individual differences in anatomy map to subjective experience? Design a study, read primary literature, talk to clinicians, build models, and when you write or teach, tell the story of these organs in full, their biology, their social life, their variability, and dignity. There's nothing lurid in curiosity, only the chance to replace myth with knowledge. Keep your questions sharp, your methods ethical, and your language generous. You will find that studying the penis and the clitoris is not just about body. It's about how science, culture, and respect shape human pleasure and health.